Welcome to this AxoCells ALS Toolbox launch presentation, presented by Axol Bioscience. Today we are launching the AxoCells ALS Toolbox. This is a new set of in vitro tools to support ALS and motor neuron drug discovery. As we know, there is no adequate treatment for ALS. Amyotrophic lateral sclerosis, or ALS, is a fatal condition that affects motor neurons in the brain and the spinal cord. There is no adequate treatment or cure, driving real urgency within the biopharma and research community for better model systems to support drug discovery and research. Motor neurons are nerve cells that control muscle activity, such as walking, speaking, and breathing. And ALS causes these neurons to die prematurely. This is called neurodegeneration. Let's take a moment to learn more about iPSCs. iPSCs are induced pluripotent stem cells. The stem cells are made from mature cells donated by a patient or often a relative, and the donation can be a blood sample or a fibroblast. The sample is reprogrammed into an iPSC using technology first created by Professor Yamanaka, winner of the 2012 Nobel Prize. And through the differentiation process, the stem cells, the iPSCs, can be turned into motor neurons and other cell types. Importantly, the end cell type can carry the traits seen in the original human donor, making iPSC-derived cells more human relevant than some other cell types. iPSC-derived motor neurons can be used in vitro cell culture studies to model ALS in a dish for drug discovery studies. And when we manufacture motor neurons from different donors, different lines, we see the reproducible differences between different lines which reflect what we would expect to see in the human patient. Classical traits of ALS in vitro models include hyperexcitability, asynchronistic firing, and irregular structures seen down the microscope. To produce these models, it's the combination of the precursor cell type, the specialist motor neuron media, accelerator supplement, and growth factors that drive the generation of the functional motor neurons that we need. Thaw to assay ready is in around 10 days. In practical real-life examples in the lab, experiments can often take around 2 weeks. That's from thaw to assay ready endpoint, applying compounds, endpoint. We regularly use qPCR, MEA, and SNA as endpoint assays. Axol operates an ISO 9001 accredited manufacturing facility in Roslin in the UK. And we manufacture to and exceed ISSCR standards. We use functional testing, functional QC, as part of our manufacturing processes. So let's have a closer look at the AxoCells ALS Toolbox. We have created iPSC-derived motor neurons from 6 donor lines. 2 from unaffected donors, 1 from a C9orf carrying donor. There's actually the sibling to an ALS donor who also has the C9orf expansion. And there's also a SOD1 patient and a TDP43 patient. We've characterized each line, each motor neuron, by ICC, key markers for motor neuron expression, SNA, spontaneous neuronal activity as measured by the in-incubator real-time imaging from the IncuSite, and electrical firing by multi-electrode array. And now the whole thing gets more interesting. When we look at ALS-derived motor neurons compared to control motor neurons, we see marked ALS-like phenotype. And to look at this in full, we have the full characterization pack in the new Toolbox brochure. So in the Toolbox, this motor neurons from 6 donors, 2 unaffected, the sibling pair both with a C9orf expansion, and patients with SOD1 and TDP43 mutations. and we see consistency of these phenotypes between multiple manufacturing runs. Our motor neurons have been well validated on leading MEA platforms, including the Axion Maestro Pro. And these cells are ready in stock, available from Axol or one of our partner distributors. We offer complete outsourced services, so you can let us do the heavy lifting on your model building or compound screening. We really hope that this new toolbox will be a valuable asset as we all work together on better human disease models and effective treatments for ALS. Thank you for your time.